Hello everyone and welcome to today's live broadcast, Complete Workflow Solution from Gene to Protein Combined Highly Advanced Expression Systems with Expression Optimized Genes, presented by Dr. Stephan Fath, Manager of Manufacturing at Thermo Fisher Scientific. I'm Susie Valdez and I will be your moderator for this educational webcast presented by LabRoots and sponsored by Gibco, part of Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want. At any time you want during this presentation, just click on the green Q&A button located at the lower left of your presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of our presentation. Also, please notice that you will be viewing this presentation in a slide window. To enlarge the slide window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having problems. Please join me now in welcoming Dr. Stephan Fath. I will now turn the presentation over to him. Welcome, sir.
Well, hello. Thank you, Susie, for this introduction. Welcome everybody on the computer here from Regensburg in Germany. Once again, my name is Stephan Fath. I'm a manager manufacturing in the Biosciences Division of Thermo Fisher Scientific, and I am heading the protein service group here at the GeneArt site in Regensburg. I'm really happy to be part of this quite interesting web-based conference format, and I'm happy to add an SynBio aspect to the agenda of this conference. I would like to start my talk with a quick look at the agenda, basically show you what will be covered during this talk. I will start out with a very brief and short introduction to gene synthesis. I will introduce you to our multi-parameter gene optimization approach and basically show you data how this optimization approach actually benefits protein expression. I will also show you some data that demonstrate which molecular mechanisms of protein synthesis are affected by this optimization approach before I will end this first part of the talk introducing to another quite nice application for optimized genes. During the second part of my talk, I will introduce you to those highly advanced protein expression systems which are offered through Thermo Fisher Scientific and introduce you to our Gene to Protein service. And by doing this, hopefully I can demonstrate you how your research can benefit from combining highly advanced expression systems with expression optimized genes, and how we can help you doing this by offering a complete workflow solution from gene to protein. So anyone who has never used a synthetic gene before, those are of course double-stranded DNA constructs which are synthesized de novo completely from scratch and based only on a digital DNA or amino acid sequence. And of course they are made exclusively according to a scientist's specification. Those genes can be made more than 10 kb in length, and of course no gene leaves our house when it's not 100% sequence verified. So you really can say when we are producing a synthetic gene, we are writing DNA. And here at GeneArt Thermo Fisher Scientific, we are considered as the excellence center for writing nucleic acids, for writing DNA. And the opportunities which are offered through gene synthesis, they certainly do explain the increasing demand and the exponentially growth in this field over the last couple of years, since it is simply a very cost-effective, time and resource-saving method in order to obtain a desired DNA construct. And as I will show you, it presumably will provide you a better expression performance than any wild-type gene. So just remember yourself or imagine yourself being in the lab. You want to express a protein. So you always relied on the availability of certain cDNA templates, which often are not available at all, contain errors, simply a time-consuming process even to get the template in your own hands. And then you had to go all the classical steps from PCR amplification to sequencing. And in the end, you probably end up with a natural gene in your expression plasmid, which has a questionable performance. By ordering your synthetic gene through GeneArt, you will save a lot of time, time you can spend on your actual research in the lab. And again, I will show you, it presumably will provide you a better protein expression performance. So, yeah, key applications for synthetic genes certainly are manifold, from basic research to cell line optimization. Synthetic genes are used in protein engineering, gene therapy, drug and target validation. But yeah, during today's talk, I strongly want to focus on protein, and here especially on antibody optimization and production. And yeah, the service portfolio we are offering here at GeneArt Thermo Fisher Scientific certainly covers the whole workflow, from project design to gene synthesis to the highly expressed and purified protein. Or, if requested, of course, only parts of it, since, of course, in addition to classical gene synthesis, we can also deliver gene variants of your target up to very complex gene libraries. But again, this talk will focus on mammalian protein expression projects. So, okay, if your interest now is protein production, why should you go with an optimized gene? And one reason for sure, and this has been known for many, many years, is that, of course, codon usage is affecting gene or protein expression. So you have a genetic code which is universal. You have 61 codons coding for only 20 amino acids, and out of those 20 amino acids, 18 use synonymous codons. And those alternative codons are actually used with an unequal frequency. So just as an example, if you take a look at this codon CGT, which is coding for an arginine, this actually would be the most frequently used codon for an arginine in an E. coli expression host. However, in a human cell, this actually would be the most rarely used codon for an arginine. By contrast, for example, AGA would be the most frequently used codon for an arginine in a human expression cell, while in an E. coli cell, those would be actually the least frequently used codon. And those codon usage patterns, they really differ in each and every organism. And so apparently, one factor, codon usage, plays a critical role during gene expression, since the codon usage of your transgene always has to reflect the tRNA availability of the given expression host cell. But yes, of course, and especially in mammalian cells, gene expression or protein expression is a much more complex process, and it is actually influenced by many different factors. So once again, if you take a look at this aligned sequence here on this slide, which should represent a wild-type sequence of your gene, this sequence might not be optimal for gene expression. So what you could do now is you simply could replace each and every codon with the most preferred codon for your expression cell. But this might also not lead to success, since a simple codon back translation might introduce some additional unwanted motifs, such as repetitions or RNA secondary structures, which could be, of course, huge obstacles, for example, for the ribosomal machinery, and therefore hamper gene expression. And so the goal always is you need to find a trade-off between codon optimization and those additional motifs which might influence protein expression. And this is why our IT software engineers here at GeneArt, basically they developed an optimization algorithm which is not a simple codon optimization, but actually is an RNA optimization. And I will talk about this in a little bit more detail in just about 1 or 2 slides. Okay, so how do you solve now such a multi-parameter optimization problem? Because you can imagine this is a huge mathematical problem. So what you could try is you could do optimization in an iterative way, where basically round after round of optimization you deal with different parameters, such as in the first round of optimization you deal with codon usage, In a second round, you take a look at RNA secondary structures, and so on, which you can imagine is a quite time-consuming and tedious process. And in the end, you might not even end up with the very best solution for your gene. You could try to test all possible sequence combinations, but yeah, just imagine a gene coding for a 100 amino acid protein. This would already create such a huge sequence space with billions and billions of different sequence combinations that actually there's no computer on Earth who can handle this in a timely manner. You could try a more random approach, so-called Monte Carlo approach, but yeah, random optimization certainly implies that you will miss certain sequence combinations and you might not end up with the best solution. And this is why our IT engineers, again, our software developers, created a so-called sliding window approach. And what this approach does, it actually does test all possible sequence combinations. However, it does this in only a limited window of approximately 10 to 20 amino acids. Within this window, the very best solution is created and the very first codon is locked down. And after locking down this very first codon, the window slides along your sequence from the 5' to the 3' end, and the whole optimization process within the window is repeated all over. And by doing this step after step, you constantly grow your optimized sequence. And of course, there's also an application taking a look at an extended window so that you can ensure that you did not introduce additional unwanted motifs, which in the end might hamper protein expression again. Okay, so if we talk about protein production, and we want to talk today about the production of human recombinant proteins, and certainly this production of human proteins is an indispensable process for modern biotechnology and biomedicine. And by far, mammalian and E. coli hosts are the most prominent expression systems to do this job. The advantages for E. coli expression are obvious. It's fast-growing, it's easy to handle, and it's a quite cheap and well-understood organism. But, of course, especially for the production of human recombinant protein, it has significant limitations, such as the lack of post-translational modifications. You will never have a suitable membrane environment for membrane proteins. Overexpression of your proteins might lead to misfolding and aggregation, so you have to resolubilize your proteins, for example, from inclusion bodies. Again, a quite tedious and unwanted process, and especially if it comes to large size and oligomeric proteins, such as, for example, antibodies, you have significant restrictions. And again, talking about antibodies, you will never be able to produce an authentic human glycoprotein. So apparently producing a native protein outside its native context is quite difficult to achieve, and in the end it presumably will differ from its native form, and this is why to date more than 70% of recombinant protein pharmaceuticals and proteins which are used for vaccination, human therapy or diagnostics are currently produced in mammalian cells, since of course it circumvents all these cons of E. coli expression. Leaving us with the question, why do we then still need optimization if we produce our protein in an autologous host? And yeah, the answer to this question is quite simple, since in nature, expression is rarely optimized for maximum expression, for maximum yield, but rather for maximum regulation. And according to this, you still face significant obstacles in mammalian protein production, such as transcriptional silencing, a destabilized or a non-stable mRNA, alternative splicing events, premature polyadenylation events, or simply inefficient translational process. So apparently all problems of gene expression, they find their source in the transgene itself. And this is why we believe that gene optimization should provide and will provide the technical solution to overcome those problems. And to demonstrate this, what we did is we carried out a large-scale comparative expression study where we basically chose 50 standard human genes from the NCBI data bank, which are representing the most interesting protein classes for basic research, such as kinases, transcription factors, ribosomal proteins, cytokines, and membrane proteins. And what we did is we de novo synthesized those genes, as you can see on the right side of this slide, as wild-type genes as well as optimized genes. Of course, still both encoding for the same amino acid sequence. When I talk about optimization, I already mentioned that our optimization approach is not a simple codon usage optimization, which we talked about before, but rather is an RNA optimization. So we do, in addition to codon usage, we also deal with GC content in order to increase genetic stability. And this also promotes actually nuclear export of your mRNA to the cytoplasm. We remove any cryptic splice sites, we remove any premature poly-A sites in order to avoid a premature polyadenylation termination process. We deal with direct repeats, again increasing genetic stability, remove RNA secondary structures, which, I said it before, can be huge obstacles for the ribosomal machinery, hampering translation, and we deal with so-called killer sequences, which are often AU-rich elements, again, in order to increase genetic stability and to prolong mRNA half-lives. And what we did then is we basically transfected in triplicates those wild-type and optimized genes and compared into HEC293 cells and compared their expression levels via Western blot analysis. And as you can see here on this slide, throughout the study, the result was quite stunning since, actually the optimized constructs outperformed their wild-type counterparts in almost each and every case, and no matter which protein class or else. And the results are summarized on this slide. So what came out of this comparative expression study was, first of all, that 100% of our gene-optimized constructs did express, which is proof for the high reliability and reproducibility of protein expression in vivo using optimized genes. And this is quite worth mentioning since, for example, 12% of the wild-type genes did not express at all. We saw that 96% of the optimized genes showed at least equal or higher protein expression levels, while actually 86% indeed showed higher protein expression levels than their wild-type counterparts. Those expression levels in the study were increased up to 25-fold, and we only had 2 low performers where actually a wild-type gene performed better than the optimized genes. Both were membrane proteins, and I would like to comment on this in just about a second on the next slide. What is shown here is that actually those gene optimization effects that we observed in the HEC293 system were not restricted to the 293 system, but since the results were reproducible also in CHO as well as in SF9 insect cells. So simply by transfecting the very same wild-type and optimized transgenes into CHO cells and insect cells, again, the optimized constructs outperform their wild-type counterparts. However, on this slide, I also want to draw your attention to this vitamin K reductase complex, which is indicated by this red dotted circle, since actually this vitamin K reductase complex was one out of the 2 membrane proteins which in the 293 system showed a higher expression performance using the wild-type gene. However, as you can see on the far right, when expressed in a CHO system, this effect was reversed again, now having the optimized construct showing better expression performance. And it is speculation, but we do speculate that what we observed in the 293 system was rather a toxic effect due to overexpression using the optimized construct, rather than actually the wild-type construct showed a better performance with regard to protein expression. Okay, so next, of course, we wanted to understand and analyze which molecular mechanisms were actually affected by our optimization approach. And in order to study this, we generated two stable cell lines using Thermo Fisher flip-in technology, a cell line expressing a wild-type version and a second cell line expressing an optimized version of a cytokine MIP1 alpha. And we did those tests by using stable cell lines, since we really wanted to compare a single copy effect of a transgene in the same genomic context, rather than comparing multi-copy effects, which could lead to saturation effects when we use a transient system. And again, first of all, we compared expression levels of those single transgenes, and this was measured by a commercially available ELISA test. And as you can see, the optimized construct for the cytokine showed an almost 300% increase, or a threefold better protein expression performance than the optimized gene. And then we wanted to understand what mechanisms are accounting for this. And in order to do this, and I will not go into the experimental details here, but what we measured is, for example, by using a nuclear run-on assay, de novo synthesis rates. And what came out of these nuclear run-on experiments was that de novo transcription rates were increased by 30% using an optimized construct. We also measured mRNA half-lives by doing real-time PCR analysis, and could demonstrate that mRNA stability increased by 14% using the optimized construct. We also measured, using Northern blot and real-time PCR, mRNA steady-state levels, and demonstrated that actually those mRNA steady-state levels of cytokine MIP1 alpha mRNAs were increased by 80%. And finally, carrying out a cell-based translation assay, we also demonstrated that even the translational process was 20% more productive when we used an optimized construct. So combining all those individual aspects finally leads to this threefold protein expression increase using an optimized construct. And those experiments clearly demonstrate that indeed our optimization approach targets each individual step of protein synthesis from transcription to translation, which is quite nice results. Okay, we also could show that by using optimized genes and overexpression of a protein using optimized genes did not affect solubility or functionality, which is of course a quite common observation. We talked about this before when you have an E. coli host. So overexpression of your gene in an E. coli host might lead to misfolding or aggregation of your target and ending up in inclusion bodies. Again, quite tedious process to resolubilize your protein and make use of it. So what we did here is we tested a set of kinases, again expressed those kinases from wild-type genes and optimized genes, then purified those kinases using IMAC affinity chromatography, and then we carried out a pull-down of those kinases using substrate-coated beads. And then we could compare equal amounts of kinases that were obtained from a wild type and the optimized construct in a kinase assay, and we could demonstrate that functionality was completely the same. So there was not a negative effect on activity due to overexpression of those proteins. And finally, to finish up this first part of my talk, I also would like to draw your attention to another quite nice feature for optimized genes, since those represent really valuable tools for functional genomics. So you all know that, of course, siRNA-mediated knockdown is a quite widespread method to analyze cellular function. But the true challenge of those experiments always is to differentiate between a true cellular phenotype and so-called off-target effects, since a given siRNA might also trigger a multitude of secondary mechanisms in the cell which could lead to a certain cell phenotype. So when you do those experiments, you always have to carry out a rescue experiment where you basically restore the wild-type phenotype. And you do those rescue experiments by co-transfecting your siRNA targeting the endogenous gene together with an exogenous version of your gene in order to restore the wild-type phenotype. But those experiments were often difficult to carry out because, or due to the unavailability of siRNAs which target the endogenous gene but not the exogenous gene you want to use for the rescue. And now with optimized genes basically carrying silent mutations, you have the free choice to design your siRNA and to design those SI or functional genomics experiments. And such an experiment is displayed here on this slide. So we analyzed CDC2, which is actually the catalytic subunit of the M-phase promoting factor, enabling cells to enter mitosis. So what we did is, first of all, again, we compared expression levels using a wild type and an optimized version of this construct, and we obtained an almost threefold increase using the optimized gene. Then we carried out a FACS analysis, and what is shown here is basically on the upper left is the wild type state, where we could demonstrate that in wild type state approximately 16% of cells are in G2 phase, ready to enter mitosis. On the upper right then, we performed an siRNA-mediated knockdown of CDC2, and what we did is we induced a cell cycle arrest, having now more than 36% of cells in G2 phase, which could no longer enter mitosis. Then we performed the rescue experiment on the lower left, where we co-transfected an optimized version of CDC2 together with the siRNA, and as you can see, the amount of cells which were arrested in G2 phase dropped down to almost wild-type levels, indicating that we reversed the cellular phenotype, and that indeed CDC2 was the target of the siRNA-mediated knockdown. Okay, so by this I would like to finish this first part of my talk. This is all published data. If you want to take a look in more detail into the experimental procedures, this is a free access journal. It's published in PLOS ONE. And just to sum up this first part, we really could state that gene design and commercial gene synthesis increases overall expression success rates. It improves protein yields in general, and as far as we can tell, it has no negative effect on protein solubility or activity of the encoded protein. And again, if you're interested in cellular, in RNAi experiments, those are also valuable tools for functional genomics. Okay, moving on. This is actually now unpublished data, and just to sum this up, of course we also wanted to benchmark our optimization approach to other algorithms which are out in the field. And what we did here is we basically, again, we chose a set of kinases, optimized those kinases, and compared the expression level using our optimized genes to wild-type levels, but also to targets which were optimized by our competitors. And what came out of this study was that indeed our gene optimizer software was the only technology which showed consistently higher expression levels compared to wild-type expression levels, again demonstrating the high reliability of this optimization process on protein expression in vivo. And it was actually the only technology which did not fail which did not fail at least for one or the other gene, while our competitors' algorithms actually did fail at least for one or the other gene. So we believe we really have a quite good tool here in hands, and now we could also make benefit using this optimization approach and combine it with those highly advanced expression systems which are offered through Thermo Fisher. And with this, I would like to go over to the second part of my talk and introduce you to those highly advanced expression systems and how we can help you with your research by offering this complete workflow from gene to protein. Again, I showed this slide more or less right in the beginning, but just to mention it again, here at GeneArt Thermo Fisher Scientific, we do offer a quite comprehensive product and service portfolio from project design to the final expressed, purified, and analyzed protein. And I want to mention this because this is quite crucial for many of our customers. Everything is coming from one hand and everything is produced in-house, starting from oligosynthesis for gene synthesis to final expression and purification of your protein. Of course, we carry out our work to the highest quality standards, and as I will show you, we also have a very easy-to-order process. With our service, gene-to-protein service offered here at GeneArt, we are offering mammalian protein expression and purification using those highly advanced expression systems, namely the Freestyle and the XP293, as well as the Freestyle and XP-CHO systems. And we also do SF9, SF21, and high-5 insect cell protein production using the bug-to-bug system. We strongly focus on mammalian protein expression because, we talked about this before, these indeed deliver the highest quality proteins with the most proper functions. And as most of our customers are really biopharma and biotech customers who are interested in antibody production, those certainly are the expression systems we have to go for. I don't want to go into too much detail here, but basically what we offer is we do offer different service types where you can order your protein, for example, simply from a specified culture volume, or you can request a specified protein amount. But this always will involve a pilot service before, basically where we start out with a 1 liter culture, for example, in order to determine titer or yield of purified protein in order to deliver the specified protein amount that you as the customer are requesting. And with this, I basically would like to share some success stories that we could offer to our customers by combining those highly advanced expression systems and those optimized genes that we are using. So this first example was actually work that was carried out at a large pharma customer. So he did the protein part of the work in-house because he wanted to use his own expression system, but he came to us and he had very poor expression performance for a set of 4 different antibodies that he produced in-house. Basically, with non-existing performance. So what we then did is we optimized his heavy and light chain genes for expression in CHO cells, and when we send those constructs back to the customer and he performed protein expression in-house, he actually experienced a 22 to 57-fold increase in protein production levels. And if you consider making an almost non-expressor to a quite decent expressor, a more than 650-fold increase in protein expression. Another example, and this is actually now customer examples, which was done here in-house at GeneArt using our gene-to-protein service, and customer used HEC293 systems. So if we start with the example, the table on the lower left, customer came to us and he requested the production of 2 different antibodies where when he produced them in-house, he got poor titers of only approximately 2 mg per liter. However, the customer was using wild-type heavy and light chain genes, he was using an endogenous leader peptide, although he was already using our quite good freestyle 293 system and the corresponding expression construct. What we did then here is when we produced it here at GeneArt, we optimized those heavy and light chain genes. We replaced his secretion leader by replacing the endogenous leader peptide with an Ig kappa leader peptide, switched the expression construct, and transfected the highly advanced XP293 system. And as you can see, those expression titers were increased more than 100-fold. So this customer was quite happy after obtaining those titer results. Comparable story on the upper left. Again, customer came to us. He wanted to express a set of 6 different antibodies, and he provided us with his in-house production levels, which are reflected by the white bars. So, however, he was using, again, wild-type heavy and light chain genes, and he was using his in-house production system. What we did here at GeneArt is we optimized the heavy and light chain genes. We tested the construct still in the customer provided expression plasmid, but switched to the freestyle 293 system. And this is represented by the dark blue bars. And as you can see, for most of the antibody targets, we already observed a quite good expression level increase. We then also tested the very same setting, but switching into the expression plasmid, which is actually the plasmid recommended for the freestyle 293 system. And again, this gave an expression boost for at least some of the targets that we were testing. And then finally, when we tested those optimized targets in the highly advanced XP293 system, you can see that a significant level increase in protein production was achieved. Again, demonstrating that we here could increase protein production levels almost 50-fold for certain antibody targets. So, again, this hopefully demonstrates you how your research can benefit by the combination of our GeneArt Gene Optimizer technology with those advanced expression systems, namely the Freestyle 293 or the Gibco XP293 systems, which are offered through Thermo Fisher Scientific. And I also want to mention that those systems, of course, they are commercially available, so which guarantees you the full transferability of those processes for further in-house work if you want to do those experiments at a later time point in your own lab. And of course, if you order a protein with us, you will always get the expression construct in the respective expression plasmid as well. And finally, I also want to draw your attention to our newest launch of service, since we are also now offering protein service using the highly advanced XP-CHO system. And I am sure you will hear a lot more about the system during this web conference, since this is really a very exciting and very well system for doing protein production. So customer success story that is displayed here on this slide. Customer had, again, a panel of antibodies which he almost could not produce in-house, so very poor performers when he produced them in-house. And he wanted us to deliver him a decent amount of protein. So first of all, we tested those targets in the XP293 system, and as you can see, at least 4 out of those 6 antibody targets showed quite decent expression levels in the XP293 system of approximately 100 to 200 mg per liter titers. However, we still had 2 targets which actually showed almost no expression at all in the XP293 system. So when we then tested those 6 antibody targets in the XP-CHO system, you can see that expression levels significantly rose further, and making basically out of those 2 non-expressors quite decent expressors with titers from 100 mg to 200 mg per liter. So customer, of course, was quite happy with that. And also the quality of those purified antibodies was very good, as I want to indicate you here on the right side of the slide, since even after affinity chromatography, monomer levels of those expressed antibodies were almost above 95%. So meaning that no additional polishing steps was requested or required in order to deliver high-quality antibody protein. And of course, those systems are not restricted to antibody production since they work as well for example, Fath or other secreted epitope tag non-antibody targets. Okay, on this slide, very briefly, I just want to give you a snapshot. If you order a protein expression service, a protein production service at GeneArt, how a documentation could look like, but I also want to strongly mention that we are really a highly customized service, where you have the free choice of expression system, expression plasmid, culture volume, analytical or purification steps, which are requested according to your needs. So this would be just a look at a basic, a standard documentation, but of course this can be pimped according to your needs. Okay, how can you order protein production, protein purification at GeneArt? Please go to the Thermo Fisher website and search there for GeneArt Gene Synthesis. If you did that, please click on GeneArt Cell Lines and Proteins, and this will direct you to the actual website where you can order our service. Here you can download a very brief questionnaire where you simply can provide to us your amino acid sequence, your protein sequence, where you can choose the expression system you want to go for, where you can choose on the epitope tag you want us to use for purification, and some additional things that might be requested by you as the customer. You send them to us to a mailbox, and then usually within 2 to 3 business days you receive a quote from your respective sales manager. And this brings me close to the end of my talk. Just to sum up, again, I strongly want to point out that our gene-to-protein process is completely done in-house, providing full control for you as the customer as well as for us as the manufacturer. So there's no need of data transfer, no need of material transfer, which is very crucial for many of our pharma and biotech customers. You have the full design flexibility, starting with the project design, where you can choose purification strategy, where you can choose the expression system. If you're interested in a certain glycosylation pattern, for example, of your antibody. And of course, upstream of our protein service unit, we already have highly specialized manufacturing departments, for example, who deliver high-quality plasmid preparations to us, which we can use for transfecting your targets. We are quite flexible in expression scales, and we are very experienced with parallelized workflows for high throughput of your project. So if you have multiple targets that you want to have produced, we are very experienced doing that. And of course, as I hopefully could demonstrate you, we really use the best-in-class reagents by using Thermo Fisher XP293 and XP-CHO systems, as well as the Freestyle 293 and CHO systems. With regard to purification, we use state-of-the-art equipment, and we have constantly growing capacities of approximately 2,000 antibody purifications per year, plus additional 500 non-antibody purifications per year. And those capacities are constantly growing, and depending on your need, we can make things happen quite quickly, since we are part of such a huge organization, which is Thermo Fisher Scientific. And of course, depending on the need of your project, we also can provide you with state-of-the-art analytical data of the produced protein that you are requesting, ranging from endotoxin measurements to monomer detection of your antibody, capillary gel electrophoresis, whatever is requested. Again, we are highly customized. And so we think this is a very smart way for you as a customer to receive a purified protein in a very short delivery time. And I mentioned it before, you always also will get the optimized gene in the respective expression vector for further in-house work. And I also want to point out that during each step of your project, you will be supported by a highly experienced and highly motivated project management team, which basically will support you at every step during your project. And with that, I would like to finish my talk and basically, would like to hand over to the Q&A part if you have questions for me.
Thank you so much, Dr. Fath, for that informative presentation and such important data. We will now start the live Q&A portion of this webinar. Audience members, if you have questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on the screen and click the send button. We'll answer as many questions as we have time for. So let's take a look at our first set of questions. Dr. Fath, an audience member would like to know, how long does it take to receive a protein? What is your turnaround time?
Okay, so turnaround time, of course, somehow the complexity of your project plays a role here, and also the batch size of your project might play a role here. But I would say, or I can say, for a standard gene-to-protein project, starting from project design to gene synthesis, subcloning, plasmid preparation, and then protein production using our service, the standard turnaround time would be 30 business days.
Wonderful, thank you. And which purification methods do you offer, including epitope tags?
Again, I said it before, so we are really highly customized, and we are also happy to adopt customer protocols if they are provided. But standard projects basically involve, as a first step, an affinity chromatography step, and with regard to epitope tags, you have the full choice of epitope tags which are out in the field. But standard epitope tags we are using are, of course, his tags. We use flag purification, strep tags. We can do dual steps, basically putting in, and that's the beauty because we have gene synthesis up front and a project design up front. So you can put a N-terminal his tag, for example, and a C-terminal strep tag, and then do a two-way purification. Of course, we offer additional polishing steps, so we can do ion exchange chromatography afterwards. And especially when it comes to antibodies, and I said it before, most of our customers are antibody customers. We always include a polishing step using size exclusion chromatography. And here we have all scales of columns to offer.
Thank you. We have some wonderful questions coming in. Here's another. How much protein will I get if I order your service?
Well, this is a question we get a lot, which I obviously cannot answer per se, because the yield of your protein always depends on the target productivity, and it also depends on the requested quality that you have as a customer. I only briefly touched this during my talk, but if you request a specified protein amount, we always usually do a 1 liter pilot scale, and based on this we can determine titer. We can purify it and determine the yield of the purified protein. And based on this, you can choose if you need additional protein and want to scale up your project, or if this is already sufficient for you.
Okay.
I hope
and
that
Dr.
it answered that.
Perfect. And Dr. Fath, what is the cost? Example, like 100 milligrams or 1 gram of protein, what would that cost?
Question goes in the same direction, of course. Again, this depends on the target productivity, and it also depends on the quality of your final protein that you request. Again, we would, in case of such a project, if you request, for example, 100 milligrams, we would start out with the 1 liter pilot. And in many, if not most cases, if you want to produce 100 milligrams of an antibody, the XP293 system, for example, already delivers this amount of protein. So you're good to go with the 1 liter pilot. This pilot, if you ask me for a cost, this usually starts around 4 to 5K, I assume. But yeah, this you would have to request with your respective sales representatives. But that's the range, I would say. And then usually you already get the 100 mix out of this pilot. If you need 1 gram, this is of course difficult to say, but based on the pilot results, we can then estimate how much more culture we have to produce, and then we will have to set up a follow-up project in order to deliver this 1 gram of protein.
Thank you. In line with that, what are your capacities?
Yeah, capacities. So I really can say we are very well equipped here for our protein service, but capacities, of course, depend sort of the product mix, which is in the pipeline at a given moment. But I could say that if we are talking small-scale expressions in the range of 100 milliliters, so we could do approximately 300 antibody expressions, purifications per week at this lower scale, 100 milliliter scale. If we talk mid-sized scale in the 1 liter range, I would say we have capacities to produce 60 antibodies per week. And then, yeah, I say we can offer up to 50-liter wave production. Of course, this then capacities are getting a little bit lower for those large-scale projects, but in the 50-liter range, we can do 3 to 4 antibody projects per month, I would say.
Thank you so much. We have time for a few more questions. How do you guarantee that someone gets something for the money if they submit this with you?
So usually how we do set up our projects is that we set them up as sort of milestone projects. So, for example, if you have a completely unknown protein which has never been produced before, so we would start with a small-scale expression, which we call an expression proof, basically to determine, is your protein expressing, how is it expressing, in order to determine best harvest day, is the protein secreted, is it cellular, where we have to purify it from, and you will always, I said this during my talk, you will, during each step of your project, you will be in line with your project manager, and he will give you real-time updates on your project. And so if you're not satisfied how a project is going, you could stop at a given milestone. Actually, we don't face this a lot. But I guess the guarantee that you get something for your money is based on the fact that we always do pilot expressions as sort of a milestone project, which ensures that you don't have to pay the whole money for a complete project if we cannot produce it. for example. But this rarely happens, I really have to say.
Right. An audience member would like to know, what is part of your QC, or what is included in the documentation?
Yeah, I also touched that very briefly during my presentation. Again, it's highly customized. So I would say if you, the minimal documentation you always will receive would be an SDS page of your purified protein, a Western blot if needed, a protein concentration, the delivered amount, and if we talk antibody, for example, an HPLC analysis stating the monomer content of your antibody. But this is based on your needs. This can be extended significantly. So we certainly can do endotoxin measurement. We can do capillary gel electrophoresis in order to determine purity of your protein. So you have the free choice here. So, and this is always set up with you as the customer when we design the project.
Thank you. And do you offer membrane protein purification in this process?
Here you got me. So although we are highly customized, we do not offer membrane purification. However, we do offer membrane expression. We do not offer membrane purification because this is basically always a real research project, and yeah, we are a fee-for-service based type of service. But what we always can do is we can transfect cells with your membrane transgene, express those membrane proteins, and ship cell paste to you. And the documentation then basically would include a proof of expression, such as a Western blot. But purifying membrane proteins, no, we are not offering, unfortunately.
Thank you so much. Thank you again for this wonderful presentation. Do you have any closing remarks before we close for this webinar? We don't have time for too many other questions, unfortunately.
Well, closing remarks. Yeah, I hope I could demonstrate you how we can help you with your research by combining those our classical business model synthetic genes with those highly advanced expression systems. And yeah, if you have additional questions, I'm happy. You can reach out to me via email or else, and I will be happy to answer any additional questions if I can. And yeah, just go to the website, and again, we have a highly dedicated project management team, so you are in good hands when you're ordering a project with us. So you really get real-time updates. We can provide you with intermediate reports during the project if you want to decide how we should go on with your project, because of course you are the expert, and we sometimes cannot decide if your interest is quantity or quality of protein. So we can, if you want to, we can let you make decisions how to continue in real time during your project. And yeah, again, you can reach out to me at any time, and I hope I could demonstrate you some nice data with this talk.
Dr. Fath, thank you so much for your presentation today. I'd also like to thank LabRoots and Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. Audience members, additional questions will be answered and published on Thermo Fisher Scientific's website. You will receive an email notification when answers are available. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Enjoy the rest of your conference. Goodbye.